Start recording, share the screen. All right, can you guys see my screen?
Yeah, I can see it.
Thank you. So we're supposed to be starting chapter 6. Actually, we're supposed to start it last Thursday, but hopefully we can get started and not fall too far behind because we have only about 1.5 days on chapter 6. And then we'll be doing lab 7 in the lab tonight. All right, any questions about any of that? I need to send you the unknown number and your first results because you started this on Tuesday. And that means the gelatin hydrolysis test is incubated for a week. All right, I'll try and work on that during the lab and after the lab. And hopefully I'll get that emailed out tonight. Now remember, you guys can see my screen, right?
Yes.
Yes.
Remember, where is it? That the first communication will be over email concerning the unknown project. After that, all communications should be done in your unknown notes page, which is I think week 4, week 5 lab. There it is. So click on your unknown notes page. When I send you the information tonight, you should put in your unknown number. Put that at the very top, and then put down your observations on what you send you, as well as your conclusions on what I send you. Your observations are the results, the tube was yellow, and then the conclusion is there was acid production in the BR lactose test. Okay? Anyways, all future communication will be here, and you will have a link, which I don't have because I'm an instructor. I've got the grader link. You've got a submit link. And when you submit the page, you can submit a page. And at the very bottom of the page, if you have a test request, put the test request at the very bottom of the page. But like I said, you should have your unknown number on it, your observations, your conclusions, what your thoughts are. You should have on each day you're giving a submission, you should have what species are under consideration. For example, let me see if I can quickly get that. When you're looking at the gram positives, for example, and you're considering all the gram positives, that is still spinning, there it goes. Well, that didn't work. I don't think I have it here. It's on my thumb drive. Anyways, list the species that are under consideration. Okay, and how you can get those species under consideration is, come on, shut. It's not going to shut. How weird. Next to your unknown notes page, it's the unknown list, the different species list. And if you click on that, you'll see all the different species. This won't show very well because it's going to be in Canvas, and so it'll be very small, but there you go. I'm sure you can read that. That's all of the species. I don't know, 20 to 30 of them. All right, any questions? If not, shut that down, shut that down. Oh, that did shut finally. Let's go to chapter 5, microbial metabolism. Hopefully we're near the end. Yeah, we finished talking about comparing aerobic respiration with anaerobic respiration with fermentation. And we talked about this table. Any questions about anything? So apart from glucose metabolism, your body can metabolize any carbohydrate, and it'll generally break the carbohydrate down into glucose or convert it into glucose. Have we talked about metabolism yet? Nobody's answering that question, so we can convert one sugar to another, and all organisms will typically convert other sugars to glucose and then metabolize glucose. And the same true with any carbohydrate. They'll convert that or break it down into simpler sugars and then convert those simpler sugars to glucose if it isn't glucose to begin with. There is a problem for large molecules, and that is they cannot cross the cell membrane, but cells, including your own cells of the gut, can digest like large carbohydrates and other large molecules by secreting an enzyme extracellularly, digesting the carbohydrate outside the cell, and then bringing in the smaller monomers like glucose into the cell. And that's true for bacteria too. Besides using carbohydrates, cells can also metabolize lipid and protein. Obviously, they have a trouble with digesting these large molecules, but they will secrete extracellular enzymes, break them down into the monomers, and then bring the monomers across the cell membrane. We did talk about humans can digest lipid like a marathon runner will, but they don't do that initially. They have to be 45 minutes into their run before they will start digesting lipids. How microorganisms catabolize lipid and proteins as they produce lipases and proteases as extracellular enzymes that break down the lipids and proteins into the monomers, and then they're brought in. The glycerol can be brought into the Krebs cycle, and some of the fatty acids, not fatty acids, some of the proteins can come into the Krebs cycle. Some of the proteins can go into glycolysis and then through the Krebs cycle. The fatty acids are cleaved into 2 carbon units, and then they enter the Krebs cycle directly. All right, any question about any of that? Proteins are a little more difficult to metabolize. You have to remove the amino group, the ammonia amino group, and then that will go into the blood, which will be collected by the kidneys, and then that will be put into the urine. So you have to remove the amino group, and then you can metabolize the remainder of the molecule, depending on what the protein is and what the amino acid is, where it will come up. Anyways, we break down proteins into amino acids, lipids, that's what that's for. It got put over here, that should be a lipid right there. Will be broken down into organic acids and then broken into 2 carbon chunks in Krebs cycle, as well as lipids will be broken down into glycerol, which will go into glycolysis. Oops, too far. This graph is showing you the interrelationship of carbohydrate, lipid, and protein catabolism. So proteins will be broken down into amino acids. Let me blow this up. And then depending on the amino acid, it will come into the glycolysis someplace, or it will come into the Krebs cycle. For lipids, the glycerol will enter the glycolysis. The fatty acids will be cleaved into 2 carbon units and be converted into acetyl coenzyme A and then enter the Krebs cycle. And carbohydrates will be converted into sugars, which will be converted into glucose, and then enter the aerobic respiration with glycolysis. All right, any question about any of that? I've already mentioned that the oxidation of glucose in aerobic respiration is the most efficient. This is the most efficient way an organism can metabolize nutrients and then make ATP. The ATP can then be used for various cellular processes, like the biosynthesis of molecules. Now, I've talked about anabolic and catabolic pathways as being distinct and separate, but in reality, they have common intermediates, and they are not separate pathways. We call amphibolic pathway a metabolic pathway that can function in both directions, meaning where you can catabolize a molecule, but if the reaction can be reversed, you can synthesize the molecule. Any question about any of that? Amphibolic pathways are a bridge between carbohydrate, lipid, protein, and nucleic acid metabolism. Wherever we have a, let me see if I can blow this up, a double arrow, that means it's an amphibolic pathway, and the reaction can go either way. This is in our cells. Green plants are much more efficient, and they have a lot more reactions that can go either way. But as you notice, most of the Krebs cycle and glycolysis, you can go both ways. And sometimes it's not showing that the reaction is, what do you call it, going both ways. But I think everybody knows if you eat too much carbohydrates or glucose, you're not going to store that down as glucose or carbohydrate. You convert that into glycerol and fatty acids, and then make lipids, okay? And then store that down as lipids. Any question about any of that? So yes, when we eat too much carbohydrates or protein, I don't see that here. There's amino acids there. So we have the protein broken into amino acids. Those all, either the protein or the carbohydrates can be converted into lipids, and then we'll store it that way. And then vice versa, you can eat lipids and it can be metabolized. Most of the time for a couch potato, you will not burn the lipid directly as a lipid. You will convert it when you eat a lot of lipid. You will convert it into acetyl coenzyme A, and then most of that will be converted into glucose. And then when your body needs ATP, it'll send glucose through in the normal way. Any question about any of that? You know, a marathon runner can metabolize lipids, but not immediately. The point is that the amphibolic pathways integrate metabolism, And our cells are lousy at integrating metabolism. Green plants are much better, and microorganisms are much better. When they have one molecule, they can usually convert that into something they want. All right, any question about carbohydrate metabolism? It is a primary way that many cells obtain their energy needs and use the metabolism of carbohydrates to make ATP. There is another way you can make ATP that does not involve carbohydrate metabolism. And obviously there's more than one way, but we're going to talk about photosynthesis. We could talk about chemosynthesis, but I'm not going to. Photosynthesis is the conversion of light energy to chemical energy. And it could be the conversion of light energy to the energy stored in ATP or the energy stored in glucose. Chemical energy may be used to convert CO2 to glucose, and that we call carbon fixation. This is performed in green plants, I think everybody knows that, in algae, but in many bacteria, including the cyanobacteria. This is a mechanism for synthesizing organic compounds like glucose, which is essential for animals and microbes to feed on, meaning we would not have food if it wasn't for the photosynthetic organisms. And you say, but I eat beef. Well, the cow eats the grass or the green plant. Photosynthesis only occurs in photosynthetic organisms. They contain a light-trapping pigment. You actually know one of them, and it's chlorophyll. When light hits chlorophyll, the light releases an electron from chlorophyll, and then that energy in the light is converted into chemical energy using the electron. And how that's done is via an electron transport chain. We'll take a look at that in a minute. ATP and NADPH are generated, but they may not stick around because the ATP and the NADPH can be used to synthesize glucose from CO2 and water. And the synthesizing of glucose we call carbon fixation. There are actually two types of photosynthesis, and most of you have probably never been taught that. There's cyclic photosynthesis and non-cyclic photosynthesis. Most of you have been taught non-cyclic photosynthesis, where the summary equation is 6 CO2 plus 12 water molecules plus light generates 1 glucose molecule, C6H12O6, 6 oxygen molecules, and 6 water molecules. Sometimes you can remove this, or somebody who's the author will, and then we'll remove 6 water here. I prefer it shown this way because when we look at where all the 12 oxygen atoms of oxygen come from, they all come from water. Any question about any of that? You do need to know the summary equation for non-cyclic photosynthesis producing oxygen. There is another non-cyclic photosynthesis reaction, and it does not produce oxygen. So some anaerobes can engage in photosynthesis, fixing carbon into glucose. And they do use light, but they're using hydrogen sulfide instead of water. And they don't make oxygen, they make sulfide. You don't need to know that. I'm not gonna quiz you on that. However, you should know there is another photosynthetic reaction. And as far as I know, all photosynthetic organisms can engage in both cyclic photosynthesis and non-cyclic photosynthesis. Like a green plant, whenever it needs glucose, will engage in non-cyclic photosynthesis. But if the green plant needs ATP, it will engage in cyclic photosynthesis. In cyclic photosynthesis, all that's made is ATP from ADP and phosphate. Of course, you need the light. I guess that should be up there. You do need the light still. All right, let's talk briefly about cyclic photosynthesis. What happens is the light hits chlorophyll in the photosystem 1 molecule, which has a lot of molecules in it. And the electrons get stimulated by absorbing the light, and then the electrons move from chlorophyll, and they move to this protein, and they move to that protein, they move to that protein, and they return to that protein. This should look familiar to you because the electron is moving through different proteins in a membrane. Let's blow that up a little. Hopefully this looks familiar to you. Along the way, as the electron is moving, when the electron moves through this protein, it gives the protein the energy it needs to pump hydrogen ions from this side of the gradient to that side of the gradient. And then the electron returns to the original chloroplast, chlorophyll molecule that the electron started from. And that's why we call it cyclic photosynthesis. And that is the electron moves in a cycle and it returns to its starting position. Now the hydrogen gradient is a twofold energy gradient. There's more plus charges here, there's more hydrogen here. We're going to want to move from this side of the membrane to that side of the membrane. And they can only do that through an ATP synthase protein channel. And as the hydrogen ions move through this protein, it gives the energy the protein needs to phosphorylate ADP to make ATP. This should be very familiar to you because it's very similar to the electron transport chain of aerobic respiration. Any question about any of that? For eukaryotes, where these proteins are in the membrane is the chloroplast membrane, the inner membrane of the chloroplast, okay? For prokaryotes, since they don't have a chloroplast, this membrane will be the cell membrane. And on a prokaryote, where they're pumping the hydrogen ions is outside the cell into the cell wall. In the chloroplast, where they're pumping the hydrogen ions is between the inner chloroplast membrane and the outer chloroplast membrane. All right, any questions about cyclic photosynthesis? So when we look at the requirements for ATP production, we can notice that the energy source can vary. For aerobic respiration, we're using mostly glucose or some organic chemical molecule. And we didn't mention it, but the chemosynthetic organisms can use an inorganic chemical molecule like sulfur or ammonia. In both cases, the electrons are stripped from the molecule, and then the electrons are used to make ATP. mainly in the electron transport chain of aerobic respiration. But for photosynthetic organisms, we use, or they use sunlight, which also causes electrons to move. And then the electron movement can be used to make ATP in an electron transport chain right here. So the electron source can vary, and the electron acceptor can vary. For photosynthesis, the electron acceptor is electron donor, meaning the chloroplast. For aerobic respiration, the electron acceptor, the final electron acceptor, is oxygen. For anaerobic respiration, it's some molecule other than oxygen. And then for fermentation, it's an organic chemical compound in the process of respiration, glycolysis and fermentation. All right, any question about any of this? If not, I'm going to close that down and begin chapter 6. Was there a question? Now remember, it's 6 o'clock. Somebody mentioned something if I don't stop. All right, chapter 6, microbial growth. You should know the terms. We'll talk about each of these. You should understand the various requirements for growth. There's both chemical and physical requirements. You should understand the 4 phases of growth and be able to compare the different methods for measuring microbial growth. Any question about the major goals and the rough outline? You can tell I'm moving quickly because we're a little behind. Microbial growth. When we're talking about microbial growth, we're referring to an increase in the number of cells, not in the cell size. I used to teach middle school students, and when we were talking about them growing, hopefully we weren't talking about them increasing in number. Okay? That's meant a little bit as a joke. Let's watch a little video on time lapse for growth. There are various requirements for microbial growth. There are chemical requirements and there are physical requirements.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting it. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, could you guys hear that?
Yes.
Yeah.
Good. Okay, so some physical requirements include an organism has to be at the correct temperature, the correct pH, the correct salinity, correct osmotic pressure. Some physical requirements for organisms to grow. They have to have a source of carbon, a source of nitrogen, a source of sulfur and phosphorus. They have to have a source of trace elements. They have to have the correct oxygen environment. And if they need it, they have to have the organic growth factors. When we're talking about an organic growth factor for us, vitamin C would be an organic growth factor. We need to obtain that from our diet because we cannot make vitamin C. And some microbes have organic growth factor requirements, like Streptococcus pyogenes has organic growth factors that you have to add to the media. And I don't know what they are, but if we supply red blood cells, it will provide the organic growth factors that Streptococcus pyogenes needs, because we cannot grow Streptococcus pyogenes on a general purpose media, meaning something like tryptic soy agar. We need a special media in order to grow it in. And then there's some species which they've tried for like over 100 years to get it to grow in culture, but we've never figured out what the cells require in order for them to grow. Syphilis would be an example. People have been trying to grow that for over 100 years. And then another one, I'm losing what the heck it's called, but it's the bacteria that causes cat scratch fever. I'm not remembering what that organism is, but the organism that causes that disease, that organism, we cannot grow it in culture because we do not know what its organic growth factors are. Of course, maybe what somebody needs to do is grind up cats and then supply that to the petri dish. That organism grows on cats, and that's why we call it cat scratch fever. Any question about any of that? All right. When we're talking about physical requirements, all organism has a temperature requirement. Each species has the minimum temperature it can grow at, the maximum temperature it can grow at, and the optimum temperature it can grow at. You can classify microorganisms into groups on the basis of their preferred temperature range. The psychrophiles are the organisms that like growing at cool temperatures, around 10 degrees Celsius. This is the temperature of your refrigerator. Okay, and I think we mentioned, no, we haven't yet, that after some food that doesn't have preservatives in it, after you've had it in the refrigerator for a week, you should throw it away because the psychrophiles can grow in your refrigerator and then spoil the food. Any question about any of that? The psychotrophs are organisms that grow a little bit warmer than a psychrophile, but not as warm as a mesophile. The psychotrophs might be organisms that grow at room temperature. And for the life of me, I can't think of anyone that does that. Various soil microorganisms are psychotrophs. The mesophiles are organisms that grow best around 37 degrees Celsius. All of the human pathogens are classified as mesophiles. And that's why they're mesophiles, because they grow best at our body temperature. And that's why they're a human pathogen. The psychrophiles cannot grow in us. We're way too warm, and so they wouldn't fall apart. Their enzymes would move into the wrong folding pattern and wouldn't work. And so the psychrophiles and all the other organisms cannot grow in us, only the mesophiles. And the mesophiles cause human diseases. Any question about any of that? Okay, then there are organisms that grow best around 60 to 65. These we call thermophiles. And if you ever look at the chicken going around in a rotisserie at Safeway or some other grocery store, and you notice it looks a little dry and that it's probably been there a long time, the thermophiles can grow at that temperature. They're not going to grow in you, but they can grow on that food, and they can cause the food to spoil. Now, generally, they would take about a day to cause the food to spoil at that temperature, maybe 2 days. And usually the chicken, it'll be sold before that time. But if it looks old, you might not want to buy it. The hypothermophiles were something that wasn't known when I went through microbiology. These are organisms that grow best above 90 degrees Celsius. Remember that 100 degrees Celsius, which is near the optimum, is the temperature of boiling water. Now, when we say an organism is a mesophile or whatever organism we're talking about, the optimum and the range are not highly defined. Each species will have its own optimum and its own range. For example, most human pathogens do grow best at 37 degrees Celsius, but Staphylococcus aureus, when it's growing on the skin, or Micrococcus luteus, which was only found on the skin, grow best at around 30 degrees. And that's because the skin is a cooler temperature than inside the body. And so Micrococcus luteus optimum is not 37, it's actually 30. The point is that each species has its own optimum and its own range. Any question about any of that? Food spoilage can occur rapidly whenever the food is kept between 60 degrees Fahrenheit to 130 degrees Fahrenheit, which would be something like 15 degrees Celsius to 52 degrees Celsius. In this temperature range, the food spoilage organisms may grow quickly. And if they produce toxins, you will quickly get the toxin made, and then the food can cause people to be sick. Now, above that temperature and below that temperature, about down to minus 40 and up to, 150, microorganisms can grow, but only slowly. And I'm not sure what would be an example. A refrigerator that's too warm could be at 45 degrees, for example, and the bacteria can grow, just slower. Anyways, if the food is kept at the wrong temperature for too long, food spoilage may occur. And that could be happening even when you refrigerate it. If you have it in too large of a batch, the food is in too large of a batch, it doesn't cool quick enough. Whereas if you have it in a smaller batch, it'll cool quicker. And then you prevent the growth below 15 degrees C. All right, any question about food spoilage by mesophiles? If not, let's move on to other physical requirements. pH. Most bacteria grow around a pH of 7. So we can say between pH 6.5 and 7.5. When you leave that pH range, most bacteria will have a slower growth or will not grow. Lab cultures, when they grow in a media, they tend to metabolize that media and produce acidic microbial products. The acidic microbial product can inhibit the growth. And we actually use that, like when we're making sauerkraut. Well, what's another one? Maybe cheese. Not a whole lot of things can grow in these and cause them to spoil because of the lower pH. To minimize the problem with the media that we have in the lab, because when organisms grow in it, they'll make acidic products, and that will inhibit the growth of the microorganism of interest. So to minimize that, we put pH buffers in the culture media. And then that, what I said, modifies the sharp drop in the pH. And so it keeps the pH closer to the normal pH around pH 7. Now there are organisms that prefer growing at a lower pH, like molds and yeast. They don't like growing around pH 7. They actually prefer growing between pH 5 and 6. And then there's some bacteria that grow in acidic environments, like between pH 0 and pH 3. Your stomach is around a pH 2. And I think we mentioned that Helicobacter pylori, which grows in your stomach, is an acidophile, and it likes growing in an acidic environment. Anyways, the term acidophile means it grows in an acid environment. Any question about any of that? Not hearing anything, so I'm moving forward. Osmotic pressure is also a requirement of microbes. We can put salt on things or high sugar content, and then create a high osmotic pressure, which will remove water from the cell, preventing microorganisms from growing in the cell. Why you can keep molasses, salted meat, like beef jerky around, even if it's open, or peanut butter. High osmotic pressure removes water from the cell, and then the cytoplasm will shrink. It's called plasmolysis as the water leaves the cell. That can kill a cell if it's sensitive to the loss of water, like E. coli would be killed if it fell into molasses, for example. And we can use this to preserve food, like candied fruit or even dried fruit. What you're doing with dry fruit is moving the water, and that increases the osmotic pressure. We salt fish to help preserve the fish. And obviously beef jerky is both low water content as well as high salt content, which preserves the meat. Any question about any of that? Halophiles are organisms that like growing in salt. An obligate halophile requires a high salt concentration to grow, like some of the organisms growing in the Dead Sea between Jordan and Palestine are obligate halophiles. You take them out of the high salt concentration and they'll die. A facultative halophile is one that does not require high salt concentrations to grow, but it can tolerate salt concentrations greater than 2%. Some can grow at 15% or higher. For example, Staphylococcus aureus is a facultative halophile. It can grow on a TSA plate, but it can grow on your skin, which can get up to 20% sodium chloride. In a hypotonic solution, low osmotic pressure, meaning a lot of water around, the water can enter the cell by osmosis. And if the cell doesn't have a cell wall, as water continues to enter the cell, the cytoplasm will expand and expand until the cytoplasm bursts, meaning the cell membrane bursts, and that'll leak out the cytoplasm, and that'll kill the cell. But if you have a cell wall, the cytoplasm can only expand up to the size of the cell wall. And then the cell membrane can't expand any further, and so no more water can enter the cell. And if you put a green plant cell in a bucket of water, or a beaker of water, water will enter the cell until the water expands the cell membrane up to the cell wall, and then the cell doesn't die. The cell wall will protect the cell. But if you were to put your own red blood cell into the beaker of water, water would continue coming into the cell until the cell bursts. Any question about any of that? Another chemical requirement for life is carbon. Typically, cells have about half of their dry weight from carbon. Dry weight means we removed all the water from the cell, and then we weigh the cell. Half of the weight will come from carbon in the molecules of the cell. Obviously, the majority of the weight of a live cell will be water. If you remove the water, half of the weight remaining is carbon. Carbon is used in structural organic molecules. It's used as an energy source. Some cells can use carbon directly as CO2 from the air or the water. And these we call autotrophs, cells capable of using CO2 for their carbon needs. Other cells we call heterotrophs, and they get their carbon from an organic carbon source. For example, humans are heterotrophs, and we get our carbon from an organic carbon source, like my drink here. It's a fruit punch, and the carbon would be, I don't know, fructose in the drink. Cells also have a nitrogen, a sulfur, and a phosphorus requirement. All cells have to have these elements. Nitrogen is 14% of the dry weight of the cell. We find nitrogen in amino acids, obviously proteins, DNA, RNA, and ATP. Many cells can obtain their nitrogen needs from decomposing proteins or possibly decomposing amino acids and then getting their nitrogen needs. But many bacteria can get their nitrogen from a nitrogen compound like ammonia or ammonium, nitrate, or other nitrogen sources. A few bacteria can even fix nitrogen gas in the air, and these we call nitrogen fixing organisms, and they engage in nitrogen fixation, pulling down nitrogen from the air and then making it available to other life forms once they pulled it down. And the cell, of course, dies. Cells have a sulfur requirement. You can get sulfur from amino acids. Thiamine, that's a B vitamin. Biotin, another B vitamin. We use sulfur in making some amino acids like methionine and cysteine. We have sulfur in DNA and RNA. Sulfur often is obtained by decomposition of proteins. It's mainly where we get our sulfur from. But green plants and some bacteria can obtain sulfur from sulfate ions or hydrogen sulfide gas. Like E. coli can get it from sulfate ions. Phosphorus, another chemical requirement of all living cells. We need phosphorus for making DNA, RNA, ATP, and then phosphorus is needed in the cell membranes and other membranes because it's a part of phospholipid. For most bacteria, they will get their phosphorus needs from phosphate. an important source of phosphorus. I think we can even use phosphate, although we prefer getting our phosphate from the food we eat and the liquids we drink. And we can metabolize phosphate from breaking down like cell membranes as well as nucleic acids. All right, any question about the chemical requirements of living cells? If not, let's talk a little bit about trace elements. These are inorganic elements required by microbes in small amounts. The microbe usually gets trace elements from their environment. In fact, we get our trace elements from our environment, the food we eat and the liquid we drink. A trace element for us would be something like copper, magnesium, elements we need in trace amounts for our cells to function. They're often required as an enzyme cofactor, like I think magnesium helps out as a cofactor in aerobic respiration. And here's a few other trace elements: iron, copper, zinc, how do you say that one? Molybdenum. All right, any question about trace elements? Organic growth factors are required if the organism requires an organic chemical compound. You get them for a bacteria from the environment. We get ours from the food we eat and the liquids we drink. This is an organic molecule that the organism needs, and we cannot, or they cannot make it. Examples for us would be certain vitamins like vitamin C, certain amino acids, we call it an essential amino acid or an essential fatty acid. And then purines and pyrimidines. We need these in our diet, or DNA and RNA, because we cannot easily make purines and pyrimidines. I think we can make purines and pyrimidines from folic acid. But other than that, we do not make purines and pyrimidines. Vitamin C is the best example, though. Organic growth factor that we need in our diet because we cannot make it. All right, any question about the various chemical requirements for living organisms to grow? Oxygen can be a chemical requirement, and all organisms have a certain oxygen requirement, meaning whether they live with oxygen, or they live without oxygen, or they live with reduced oxygen. Molecular oxygen is, in a sense, a toxic substance, and we are bathed in it, and oxygen directly is not toxic. But when oxygen is metabolized, it can be toxic. And the trouble is our cells use oxygen, and so they make or metabolize these toxic oxygen intermediates. There are 4 toxic forms of oxygen. Singlet oxygen, where oxygen is boosted to a higher energy state. This oxygen is extremely reactive. And in fact, phagocytic white blood cells will make singlet oxygen and then secrete that oxygen on a cell that wants to kill. For example, a eosinophil secreting singlet oxygen against the skin of a helminth, a worm, parasitic worm. Superoxide free radical is the second of the 4 toxic oxygen forms. What happens is we have oxygen and it gains an extra electron. An electron has a negative charge, so if you gain an extra electron, the oxygen molecule will have the negative charge. The trouble is this is an unpaired electron, and so whenever this molecule hits another molecule, it will steal an electron from that other molecule, because electrons don't like to be in singlets, they like to be paired. And when the superoxide oxygen hits that molecule, you can damage that molecule. The problem is that you can damage like a carbohydrate, and then it goes on to damage a lipid, and then the lipid goes on to damage DNA. The problem is initially, the free radical, although it's stealing an electron from something else, but where it's stealing the electron, it'll make that molecule a free radical. And so that just keeps going on into the cell, and that can lead to damage of DNA as the molecules steal an electron from another molecule. And that process just keeps repeating. Cells try to get rid of superoxide free radicals because they're reactive. And we have SOD, superoxide dismutase, which converts to superoxide free radicals and hydrogen ion into hydrogen peroxide and water. Now, hydrogen peroxide is toxic to the cell, and this is the third toxic oxygen molecule. A peroxide ion, such as right here, or hydrogen peroxide right here. Sorry, right there. No, right there. I didn't go backwards. I meant to go backwards. Here we go. Hydrogen peroxide. And so the cell has to remove peroxide anions if ever the cell is in an oxygen environment, because peroxide anions can get made as an intermediate in metabolism of oxygen, as well as SOD will convert superoxide free radicals into hydrogen peroxide. Fortunately, our cells have catalase, and this is one way that cells can remove hydrogen peroxide or other peroxides. And catalase will convert hydrogen peroxide into water and oxygen, removing the hydrogen peroxide. Another way that some bacteria can remove hydrogen peroxide is to, in the presence of peroxidase, add 2 hydrogen atoms to the hydrogen peroxide to make 2 water molecules. We'll look at catalase later in the lab. Any question about any of that? The fourth toxic form of oxygen is the hydroxyl radical. This is similar to the hydroxide ion, but that will be OH negative with a negative charge. The hydroxyl radical is OH with the free electron. And the trouble with the hydroxyl radical is one electron doesn't like staying one electron. It'll bump into another molecule and steal the electron because the electrons like to be in pairs. Okay? And actually the hydroxide ion with a negative charge has 2 electrons in a pair. So if we just had OH, there's no charge on it, and you have an extra electron, OH will have a negative charge. Don't confuse the hydroxyl radical with the hydroxide ion, OH negative, or the hydroxyl group like in an alcohol, the OH part for another molecule. Because these are fine in the cell, but the hydroxyl radical is not. And the trouble is, hydroxyl radicals are made by aerobic respiration in trace amounts. And if they get out into the cell, they can do damage to the cell. Hydroxyl radical is also made by ionizing radiation. And then when certain metals react with hydrogen peroxide. How cells can control hydroxyl radicals is they have antioxidants which will bind with the hydroxyl radical and then remove the hydroxyl radical. Two antioxidants which bind with hydroxyl radical in your cells happen to be vitamin C and vitamin E. And they are antioxidants. And it's a good reason to eat vitamin C and vitamin E in your diet to remove these hydroxyl radicals. All right, any question about the 4 toxic forms of toxic oxygen? Remember, we are an aerobe, so we do require oxygen. And oxygen itself is not toxic, but the various metabolic intermediates can be toxic. All right, any question about any of that? Let's finish talking about the oxygen requirements and then we'll break. Oxygen, most organisms have a requirement for what their oxygen needs are. An aerobe is an organism that requires oxygen to make ATP. And so an aerobe, at least a, let me pull that to you. Drawing a blank there. Obligate aerobe requires the presence of oxygen or it cannot grow. We can look at the requirements of oxygen by growing the cells in a media where it has different oxygen in the media. And the media I'm talking about is thioglycolate. Here we're seeing thioglycolate tube. When you make the thioglycolate tube in the autoclave, thioglycolate will react with oxygen and remove all oxygen from the media. The autoclave heats it up, and so that tends to remove oxygen from the media too. But as the media cools down, oxygen from the air will re-enter the thioglycolate tube. So at the top of the tube, we'll have full oxygen. But as you go down the tube, you'll have less and less oxygen until the very bottom of the tube, you'll have essentially no oxygen. And so when we make thioglycolate media, we will have tubes having a concentration gradient of oxygen. At the very top, we'll have the full oxygen, 20 to 21% oxygen. And as you go down the tube, there will be less and less oxygen. Till the bottom of the tube, there's no oxygen. If we find microorganisms only growing in the bottom of the tube, these are obligate anaerobes. They cannot grow in the presence of oxygen because they find one or more oxygen intermediates to be toxic. So oxygen to them is toxic. Okay? And they will only grow in the bottom of an obligate, of a thioglycolate tube. Now for the aerobes, you'll notice that they only grow at the top of the tube. And as you go down the tube where there's less and less oxygen, you'll see less growth. And then at the bottom of the tube, which has no oxygen, you'll see no growth. Why we have most growth at the top is because this has full oxygen, 21% oxygen, and aerobes grow best with oxygen. So they'll grow best here, and as you go down the tube, you'll get less growth. Now that's similar to a facultative anaerobe, This is an anaerobe, but it's facultative, meaning it can perform aerobic respiration. And in fact, if oxygen is around, it will use aerobic respiration because that's the most efficient at producing ATP. So for a facultative anaerobe like E. coli, you'll see the most growth on the top of the media because that's where the most oxygen is. And as you go down the tube, you'll get less and less growth because there's less oxygen here, so you see less growth than right there. There's less oxygen here, and that's not apparent here. There's less oxygen here than here, so you get less growth. And then at the bottom, there's no oxygen, so you'd have less growth than here. And why you're seeing less growth is because aerobic respiration is the most efficient. This is where the cells will grow the fastest. Did I mention that facultative anaerobes prefer aerobic respiration, but they don't require it? They can switch to either anaerobic respiration or fermentation and continue to grow as you go down the tube. Another type of anaerobe is an aerotolerant anaerobe. They are anaerobes, meaning they do not use oxygen in respiration. However, they have all the enzymes they need to detoxify the toxic oxygen metabolites. So they can grow in the presence of oxygen, but they don't use oxygen, so they can grow in the absence of oxygen. And this means that an aerotolerant anaerobe can grow everywhere uniformly in the tube. Any question about that? The last group are the microaerophiles. They are aerophiles, so they have to have oxygen in order to grow. However, they don't grow best at full oxygen, 21%. So we find a microaerophile growing best someplace in the middle of the tube. They don't grow at the bottom of the tube as well because there's no oxygen in the bottom of the thalloglycolid tube. Any question about any of that? Does anyone know where you can find a microaerophile? I've actually mentioned it. Helicobacter pylori, which grows in your stomach, is a microaerophile. The stomach is largely closed from the sphincter, and the bacteria in your gut in your stomach, use the oxygen in your stomach, and that reduces the oxygen in your stomach to lower levels than 21%, but higher levels than 0%. The Helicobacter pylori grows someplace best in the middle of the tube because it's a microaerophile. All right, any questions about any of this? If not, we'll stop here and I'll see you at 6:30.